Well, good afternoon, everyone. Thanks so much for joining us for today's presentation in the 2021 CCOVI Lecture Series. My name's Sarah Ackles, and I'm the Marketing and Communications Officer at Brock's Cool Climate Enology and Viticulture Institute. I will be moderating today's lecture for you. As a reminder, we will still have a question and answer period at the conclusion of the presentation today. So please type your questions into the chat feature at any time, and they'll be shared with our speaker at the end. I'm very pleased today to welcome Dr. José Ramòn Urbez Torres as our speaker. José is a research scientist at the Agriculture and Agri-Food Canada Summerland Research and Development Centre in British Columbia. He also serves as an adjunct professor in biology at the University of British Columbia's Okanagan campus. His education includes a background in viticulture, enology, and wine marketing, and he completed his PhD in plant pathology in 2009 at the University of California, Davis. José studied the diseases of woody perennial crops for more than 2 decades. His current research focuses on the development and implementation of sustainable management strategies for fungal, bacterial, and viral diseases of Canadian grapevines and tree fruits. He's also published more than 60 peer-reviewed publications and book chapters, and is the current chair of the International Council on Grapevine Trunk Diseases. Drawing from that extensive experience, José is going to share some of his research with us this afternoon. So with no further ado, let's go ahead and get started. José, please go ahead.
Thank you very much, Sarah. Share my screen. Okay, hopefully you can see my screen. Sarah, can you confirm you can see my screen and you can hear me?
I can. You sound great. Screen is up. Please go ahead.
Thank you very much. Good morning here in British Columbia. Good afternoon over there. It's my pleasure to present this lecture, and thanks again to Brock University and CCOVI for the invitation. So today we will be talking on grapevine trunk diseases. So the main focus of my talk would be first an overall introduction of what are these diseases, the different pathogens they cause, as well as the symptoms that you can see in the vineyard. Why do we care about these diseases? Why they are important, and the economic impact. A very important point I will address and highlight here is to really understand the different source of inoculum of these pathogens, what we call the epidemiology. We will give an overall overview on the current status of the trunk diseases in Canada. And at the end, we'll finish, of course, with the most important thing, which is how we can manage, these diseases. So to start, grapevine trunk diseases are caused by many different fungi. The majority of these fungi are in the Ascomycota group, but as we will see later, some fungi belong to the Basidiomycota and play an important role in some specific disease. These diseases infect grapevines through openings and wounds. And as you can imagine, there is one thing we do every year on grapes, and it's pruning. So you can see that pruning wounds are the main point of infection for these diseases. However, wounds caused by other type of practices like mechanical damage, mechanical pruning as well, retraining can be a source of entrance. Some grapevine trunk disease fungi are thought to be latent pathogens, and I will explain later what this means. But basically, some of these fungi can stay in the plant without causing any damage, but there is some trigger that can make these pathogens become virulent. Overall symptoms include either a slow or rapid dieback, and that will depend on the fungi involved on the diseases, or a sudden vine collapse. We can see usually a progressive death of the plants. So we will see, as here in the picture, either a spur, so the cordon starts slowing dieback, and of course, cause eventual death of the plant. Grapevine trunk disease occur wherever grapes are grown. There is no single grape growing region that trunk diseases hasn't been reported. However, it's important to note that the type of disease and the prevalence of the pathogens may vary depending on the geographical region. So starting with grapevine trunk diseases, we have 2 major groups: trunk diseases that affect young vineyards, and we can establish here a young vineyard within the first years after planting. So we have two diseases, Blackfoot and Petri disease. These two diseases were thought a while ago to be two separate diseases, but actually right now are considered a complex, and we call it a Jan-Vine-Decline complex because both diseases and the pathogens they cause this disease can be found in the same plant. Blackfoot is caused by the only group of fungi that are soil-borne within the Tran disease complex in the Campylocarbon, Paleonectria, Dactylonectria. Petri disease are caused by mainly Phaemonia chlamydospora, many different species of Acremonium, being minimum the most important or most prevalent, and Cadophora luteolibacea. In mature vineyards, we have what we call the canker diseases, as Botryopsphera dieback, Eutypa dieback, Phomopsis dieback, causing this characteristic, perennial cankers in the vine. But we also have ESCA, which is a very complex disease, as we will introduce later. So in terms of Jan-Vine decline, most of the symptoms associated with this disease are this poor growth, poor vigor of the plants, very slow growing in the spring, short internodes, and even when the plants demands the highest water needs during the summer months, we can see this sudden collapse. However, as you can see, these symptoms overall can be very difficult to differentiate with other causes, like, for example, winter injury, spring frost, nutrient deficiency. So it's very difficult to assess Jan-Vine decline just based on observation in the field of the plants. However, internally, you can see these fungi cause very characteristic symptoms, all this streaking of the bud. We can see here necrosis of the xylem vessels, this plugging, sometimes with tylosis, which is metabolites produced by the vine. So these are the symptoms that we usually see with Jan-Vine decline complex. There is a large range of different fungi causing these diseases. As you can see here, a list of them. In red, I just highlighted the most prevalent and the most commonly found, Phaemonia chlamydospora, minimum in the group of Cadophora, Botryopsphera, or Blackfoot. But just to give you an idea, the complexity of this disease with so many fungi involved, as you will see later happen with many other Tran diseases. ESCA is another disease now in mature vineyards, and this is probably the oldest disease we know, not only Tran disease, but probably the oldest disease we know on grapes. It's characteristic because it has very distinctive foliar symptoms that we call leaf tiger stripes. You can see here on red variety, this is a Cabernet Sauvignon. You can see this intervenial redness, or for example here a Sauvignon Blanc, how this intervenial chlorosis and necrosis of the edges. So this is very characteristic on ESCA disease. Also, we have a syndrome happening mostly during the summer months. Again, highest demands of water for the plant, that we can have a sudden collapse, and is known as apoplexy. The fungi that cause these diseases, some of them have some toxins that they produce, what we know the black missile symptoms in the fruit. This is a table grape, Thompson seedless, with these black spots caused by the toxins of these fungi. As you can imagine, table grapes are not marketable in this stage, and we also see some of these symptoms on wine grapes. Internally, one of the most characteristic symptoms is this soft yellowish bud. It is soft bud on the cross section in trunks or cordons, and this is mostly, as I mentioned before, because not only the fungi that cause Petri disease are involved, but in the case of ESCA, there is also Basidiomyces. There is a large group of different Basidiomyces. This is the mushroom type fungi that they can cause, the typical basidiocarps on the vines, and they cause this specific symptom as they feed on the lignin, which is the strongest part of the plant. Another disease we are going to talk is Eutypa dieback. This is a very common disease in many regions, but especially with high incidence in France, Australia, or California. This disease can be identified in the field because of the foliar symptoms. We can see here causing short internodes, cupping of the leaves. Sometimes the leaves get necrosis at the edges. And it's common to see that some parts of the vine may be affected with the symptom, while the other parts of the vine are still healthy. So this is mostly the infection will happen in any of these pruning gums. The canker is traveling downwards through the plant, so all this part of the plant is affected while the other can still be healthy. Characteristic internal symptom, these perennial wedge-shaped cankers. Historically, we knew that U-type palata was the main pathogen causing U-type Dieback. However, in the last years, we have, again, another very large range of different fungi associated with the disease. However, U-type palata is still the only fungus that has been proven to cause these foliar symptoms. These are just magnification of the fruiting bodies, these ascocarps, flash shape that they are embedded in the infected bark, where we can see the spores, inside, and we will go in more detail later. Another disease, Botrytis Dieback, one of the most recently described diseases. Again, you can see the many different species associated with Botrytis Dieback. Most important, the Neophysococcus and Lasiodiplodia group, because they are the most virulent. However, species in the Diplodia group, they are the most prevalent. But mostly Lasiodiplodia and Neophysococcus, they are the most virulent among all the grapevine trunk disease fungi. Different with U-type Dieback. With Botrytis Dieback, we don't see really any characteristic foliar symptom. We can see, the dieback of the arms or spurs. There is basically no spring growth in the spring for spurs. In this case, it's infected here. We can see the canker colonizing most of the spurs, so we don't see any spring growth. However, in other occasions, we can see the cankers on the cordons or trunks and still see healthy growth. So it's a complex disease in the sense that it's difficult to identify in the field. Finally, we have Phomopsis Dieback, and this is a disease that the pathogen is not new, as the pathogen causing Phomopsis Dieback is Diaportium pellina, formerly known as Phomopsis viticola. As many of you know, Phomopsis viticola is the cause of Phomopsis cane and leaf spot, and that's why this disease is really prevalent in those regions with Phomopsis and cane leaf spot disease pressure. Very similar to Botrytis Dieback, it can cause these wedge-shaped cankers, and similarly, these blind spurs where we don't see any spring growth coming. These are just characteristic of the colony and the pycnidia with this cirri. These are the conidial masses that come out of these fruiting bodies, and they get spread in the environment. However, as I mentioned here, trunk diseases is a disease complex or a complex disease. It's something that is not really straightforward. We don't see usually one disease with one pathogen in one plant. And this is, very commonly observed in many grape-growing regions, a mix of symptoms, in the bud. We can have the cankers, where we can talk before, Botrytis Dieback, Phomopsis viticola. We can have this dark streaking of vascular discoloration with the Petri disease, Botrytis as well. And we can have this soft bud, yellowish soft bud where the bacillomyces sets. So it's a progressive disease that many pathogens can infect the plant through the years, and that's why some of this complexity in the plant. As I mentioned before, grapevine trunk diseases are not new. It's not something that happened suddenly in the last 10, 15 years. And as I can show here, the first studies on ESCA happened, at the end of the 19th century in France. And since then, as you can see, in different decades through the 20th century, different diseases have been identified with different pathogens. Even here, we have one of the first description of Botrytis species causing canker was actually done in Ontario in 1964. So here in Canada, we know these diseases have happened for a very long time. However, the question is why the emergence of these diseases, why we are seeing these diseases now more prevalent. Emerging disease, based on the National Institute of Health definition, we can have either have occurred previously, but affected only a small population in isolated places, or an emergent disease have occurred throughout human history, but have only recently been recognized as a distinct disease due to different factors. This is within the grapevine trunk diseases community where we think is happening. This disease has been thought to be evolving with grapes, have been thought to be as old as vine cultivation, but different factors in the last decades have made to be more prevalent. Some of the hypotheses, that why we think Tran diseases occur or major outbreaks occur since mid-1990s. First of all, big planting booms, either for renewal of varieties or in the New World viticulture, that have been planting booms where there was a shortage of material, so production of poor quality material. But mostly one of the hypotheses is also the prohibition or phase out of some of the effective chemicals controls we have, like sodium arsenate in Europe. It was effective with ESCA, or the systemic benamyl in 2001, that it was one of the few systemic products that were able to affect internally the pathogen. Also changes in viticulture practices, as we can see here. This is just a picture of my hometown in Spain. These are 80, 90-year-old Garnacha vines. Very, very low planting density, very few cuts or spur pairs made in these bush vines, compared with what we are most usually observe these days, a high density planting, BSP, a double cordon, each of these cordon, approximately between 8 or 10 spur positions. Each spur position is going to have 2 pruning guns. So we are actually really creating an avenue for infection of these pathogens. So why do we care about grapevine Tran diseases? Well, there is no doubt that they cause a significant increase in management costs, and this is just some data, from California or Australia of the cost of these diseases. Also, in the case of young vine decline, one of the major problems is, the loss of plants before you can actually get a cash return the first years until you get into full production can result in terrible economic losses. And also the difficulty to manage an uneven vineyard. And every grower knows, how challenging it is to have plants with different ages and have to replant every year vines in your vineyard to manage. So overall, it has been studied that the cost of grapevine Tran diseases is estimated in about $1.5 billion US. And this is just based on the replacement cost of 1% of planted grapevines. And as we see, in some of the data we have, this is actually underestimated. So as an example, in France, just one disease, ESCA, has been considered as a national crisis. Right now, there are over 10% of the growing area, about 100,000 hectares, economically non-productive in France, causing over 1 billion euros of losses. So you can see the significance of these diseases. So when we are talking about the sleeping giant in your vineyard, and this is very important to understand with grapevine Tran diseases, to be aware, this is the incidence of grapevine Tran disease by age of the vineyard, and these are great economic studies done by Dr. Jonathan Kaplan at California Polytechnic in Sacramento, where basically you can see here the vineyard age. As vineyard age progresses, we see the incidence of Tran diseases increase. Of course, at the same time that the incidence of Tran disease increase, our yield is going to decrease. So we have here that by year 8 or 9, we can lose a 10, 15%. But by year 12 or year 15, we can have a reduction in yield, between 50 and 70%. So that's economic loss that nobody can really support. So one of the problems we have is, awareness of the disease. And one of the problems of the disease is it takes time until we see the symptoms in the vineyard. So, we have, for example, since the first year, we start managing powdery mildew in our vineyards. In the first year we plant, we start spraying and conducting cultural practice to minimize powdery mildew. We know powdery mildew, we know it's going to happen no matter what in our vineyards, and we can see also the symptoms, of powdery mildew. The problem with Tran diseases is we may start getting our plants, infected in year 5 or 6, and we may not see symptoms until year 8 or 9. And that's when we start, maybe, not always, applying some of the control strategies, which is obviously too late because we already have the infection in our vines. It's better than nothing, to start, but, basically, as I will highlight in my talk, we need to start thinking on Tran diseases control, same as we do with powdery mildew. Start all the way from the beginning. So I want to mention and highlight a very important part on grapevine Tran diseases, and is understanding pathogen source and spread. Basically where the inoculum is coming from, and the epidemiology of these diseases. And this is going to have a very important impact on how we manage the disease. So the first source of infection, and I'm not saying just that myself, but there is very, very clear evidence that grapevine trunk diseases occur in the nursery propagation material. So these are two recent publications where a lot of this information can be found. But basically, throughout all the stages of the grapevine propagation material, from the mother rootstock or scion blocks, hydration tanks, cold storage, disbudding, grafting, there have been studies identifying infections on trunk diseases pathogens. So this is a problem that we have coming in our vineyards. So some work we have done here in Summerland first was develop a diagnostic tool to be able to assess the health status of this planting material. One of the main issues we have with grapevine trunk diseases, as you have seen, is the large amount of pathogens that are involved. So basically what this tool allowed us was to, in just one single test, being able to detect up to 72 different fungi. So we collect samples from different nurseries, and we can have here, as you see, grafted and self-rooted samples, So from grafted material, we collect samples from the root, the basal end of the rootstock, the graft union and the scion. And of course, from self-rooted, we then have the graft union. So all these samples, DNA was extracted, was run through our DNA macroarray. And this is just, an overall of the results from these two nurseries. So you can see, first, the huge diversity of different fungi that we found. We know all these fungi cause either Petri disease, black foot disease, or what we call now the germ-vine decline complex. But for example, some of the pathogens in the Eleonectria or Dactylonectria, up to 92%. So more than 90% of the plants have those pathogens. More than 70% of the plants have Botrytis. More than 60% of the plants has Phymonella. So a very large number of infection. And also it was common to see more than one pathogen in these plants. However, while we did this work, we also noticed one thing, and that's what I was mentioning before, that some of these pathogens can be, as we know, latent pathogens. So latent pathogens meaning that they may lie dormant in the wood tissue and only become virulent after usually a stress factor. That can be an abiotic or abiotic stress factor. So if that stress factor don't happen, these fungi stay dormant in the plant. So you can see here a healthy nursery plant with no necrosis at all at the base of the rootstock. We were able to identify common germ-vine decline fungi, and at the same time, from another sample, you can see the necrosis on the xylem. We were able as well to identify, even the same pathogen. So that was our hypothesis. So what we have here is, a vine throughout the lifespan of a vineyard. A vine eventually, will die because, of course, age. But what we are hypothesizing here is that if the vine is infected with one trunk disease fungi under a stress condition, it will have a premature death. And our hypothesis also indicates that if we have multiple infection with a stress condition, this vine will even die or collapse earlier. It's also important to understand a colonization threshold, and we need to know how much pathogen is needed, to really causing this premature death under a stress condition. Because there may be plants that they are, with these pathogens in the wood, but with no stress condition, with not a colonization threshold above the limit, the plants can thrive in our vineyards. So the other source of infection we have with trunk diseases, as we see, is in mature vineyards. So we have here an infected plant. In those areas infected, we can see our fruiting bodies containing the spores. These spores, under optimum conditions, they will release and infect again pruning gums and close the cycle. We can have also spores coming from alternate hosts, and many of our fruit trees or even native plant communities can cause, are infected by trunk diseases causing this problem. These fruiting bodies can be localized in very old pruning wounds. As you can see here, this is just electron microscope of one of these fruiting bodies with the neck of the fruiting body. And basically I have here is, see if I can play that. Oh, I have to get the pointer. Let me see if I can. Okay, it's not working for some reason. Let me hold on a second if I can remove the pointer. Now, okay, sorry about that. So we are seeing here in a video what is basically the end of this fruiting body I showed you. And this fruiting body is releasing the spores. These spores, in this case, is Phyclemonium minimum, causing ESCA or Petri disease, are inside these ascos. The ascos, so this sac, contains 8 spores, which is very common for ascomycetes. And basically what is happening when these sacs are ejected from the fruiting body because of a difference in pressure, the sac will break and all these spores will basically spread through the vineyard either by wind or by rain droplets, etc. Okay, so we can continue now. So many studies have been done to understand the epidemiology, around the world of these grapevine trunk diseases, and just basically as an example, basically what we are trying to do with these studies is identifying the inoculum availability in the field, where there are low or high periods of infection throughout the year. And this is just an example of work that I conducted while I was in California on Botryopsvaria spore trapping in Napa Valley in this case. So we can see here in blue precipitation. Each bar indicates the number of spore trapped through the different months, and in red you have precipitation. So overall we see in California over 60% of spore release was from late November to early February, so late fall to mid-winter, which actually correlates with the main rain periods in the region in California. Less than 20% of spores released in fall or early spring, and almost no spores, released in the summer, where, basically we have very dry conditions. So this information is critical for us to understand how to implement control strategies. So I'm going to go to the next point, which is grapevine trunk disease in Canada, quickly reviewing. As I mentioned before, we knew trunk disease happened, in Canada, and the first report actually was in Ontario in 1964. But it hasn't been until the late 2000s, where British Columbia started experiencing significant plant mortality, primarily in newly established vineyards. This era was, kind of the industry expansion after the NAFTA agreement. Important yield losses in mature vineyards planted between 1995 and 2000. So at this stage, these vineyards were getting into the 10, 15-year-old mark, and that's when we are starting to see this decline. Historically, some of these symptoms have been associated to abiotic factors like, winter injury, spring frost, or even to some of the diseases happening due to the cold climatic conditions in this region like Crown Gall. However, in BC, they were interested in investigate other possible causes, and my predecessor at Summerland started some work in 2008, 2009, looking into trunk diseases. So the first thing was try to understand the incidence and importance in BC. So we actually conduct large field surveys with a lot of number of vines inspected. Overall, the results showed that, trunk disease vines were found in 90% of the vineyards. Overall, we found around 10% of our 60,000 vines inspected showing trunk disease symptoms. We found in young vineyards up to almost 40% incidence in a single vineyard, or in mature vineyards, almost more than 50%. And we saw a high young vine decline incidence. If we see here the relationships between vineyard age and incidence, you have the vineyard, the incidence. We can see here, all this caused by young vine decline, all this very high incidence in some vineyards with 2-3 years old. But you can see here kind of a plateau, observed here in the average. This is the time that most of the vineyards are going to get infected. But you can see, as I mentioned before, most of the symptoms, we are start seeing them later, after 10, 15 years. And we have, again, the increase of incidence based basically on age. There were some vineyards we recorded with no Tran disease symptoms. This doesn't mean that they were not internally infected. As I mentioned before, some of these diseases, they don't show characteristic symptoms, but I would bet a nice glass of wine that there will be no vineyard block with more than 25, 30 years that they don't have any internal symptoms on Tran diseases. In terms of the fungi that cause, a part of this project was to try to identify. We found more than 40 different fungi, and we did that through molecular and DNA analysis. We found Botrytis and Eutypa or Eutypa-like species in mature vineyards, Phaemonella and Ilionectria species in young vineyards. We have very low incidence of foliar symptoms, and this was very interesting and different from other grape-growing regions, where Eutypa Dieback symptoms are rarely observed, as same as ESCA, which it can be a disadvantage for us here, at least in British Columbia, because we are not going to be able to identify those diseases just based on symptoms. We were able to understand the pathogenicity and virulence, and as I mentioned before, the group within the Neophysococcus was the most virulent. And again, a significant impact in nursery planting material. However, as I mentioned before, we wanted to really understand what is happening, with this asymptomatic, but still presence of fungi, this latent status of these pathogens, following basically what I was mentioning before, understanding, which type of biotic or abiotic stressors could cause this premature death. Some of the work we have done is try to develop a recently fairly new molecular detection, which is called droplet digital PCR. The main advantage versus the standard PCR is we divide our sample in many thousand, small molecules where a single PCR is conducted in each of them. And basically, we have been evaluating nursery material not only to see the presence of ascents of the main pathogens causing germline decline, but also, the concentration of these pathogens in the plant. And this is what you can observe here. This is with Phaemonella chlamydospora. Each of these columns represent a plant. So you can see each of these blue dots is a positive for a single gene detection of that target. So you can see here a significant difference in the concentration of the pathogens in the plant. Some plants with very high concentration. These are our positive and negative control. Some plants with much lower concentration of the pathogen, and some plants with no pathogen at all. So that's what I was mentioning before. It's important to understand the threshold that the plant can actually fight against, let's say, and have this border where no symptoms will happen. If we see regarding the different pathogens, well, for example, Chardonnay, we found 100% of the plants have Phaemonella chlamydospora and Cadophora tolibacea. Merlot hardened very, very similar results. In Pinot Noir, 100% of the plants in Pinot Noir have Ilionectria. But this really clear tells us that the presence of the pathogen may not result on disease development and eventual plant death. If means that having 100% of our plants, Phaemonella chlamydospora, all these plants will die in the field, I could suggest that we will not have viticulture these days. So that's why it's very, very important to understand what really means to have this fungi in our planting material. So we can see here the difference depending on the plants. We have Chardonnay in blue, Merlot in green, Pinot Noir in red. Here is the total number of copies for Phaemonella chlamydospora on different plants. So you can see, for example, Chardonnay appeared to be the plant with the highest incidence of Phaemonella chlamydospora, one plant on Merlot. But for example, Pinot Noir has very, very low incidence. So there is a significant inoculum concentration difference between cultivars. But not only between cultivars, but also, among different parts of the plant. So in this case, Phaemonella chlamydospora was mainly found in the graft union, followed by the basal end of the rootstock, and some in the scion. There was not found on the roots. We know Phaemonella is not a soil-borne pathogen, that barely or very, very rare in fed roots. So we can see here that the main infection in the graft union can come in the process, as I mentioned before, in the nursery. It may be infected in that part. So right now, I have a PhD student, Jared Rykan. Basically, we are trying to answer some of these questions. This abiotic, which type of abiotic or biotic stress factors are enhancing the disease development in our plants? We are looking into water stress, nutrient deficiency, overcropping, J-rooting, cold temperatures, or, for example, the presence of nematodes, looking into the inoculum thresholds, and also to see if AMF, mycorrhizal fungi, that, as all we know, can help to minimize the stress in plants, can also assist to minimize the stress for these pathogens. And we are actually conducting both greenhouse and field trials on this. We have been determining the epidemiology of grapevine trunk disease in British Columbia, determine vineyard inoculum presence during the year, try to understand the high and low infection periods, determine environmental factors, and critical information, to target control practices, optimum time for pruning, or manage our pruning to try to reduce, as I will mention later. So we basically have 5 of these spore trapping devices. Throughout 3 years in the Okanagan Valley, we developed a molecular assay where we were basically counting the amount of spores trapping either weekly or daily. So this is just the example of one of our vineyards for Botryopsphera. Here you have the total amount of spores per week. This is our temperature throughout a 3-year period. So very clear, you can see the peaks of the summer and winter, and precipitation. So the first thing we notice is that most of the Botryopsphera inoculum comes at the end of winter, early spring. And while there was not really a strong correlation with precipitation, we saw some correlation with temperature. So as soon as we are above these freezing temperatures, 5-6 degrees Celsius, we start seeing the first relief of spores. Usually about 5-6 weeks later, we can see the highest amount of spores being depleted in the vineyard. So if we look this information regarding, for example, the pruning period in British Columbia, which can go from basically December until end of March or April, we can see that the last part of our pruning period, and I would say where most of the growers are pruning in British Columbia, could be about now, February, March, we start getting into the period where we see the start of the inoculum in the field. However, we can see here that growers starting maybe in December or January, they may be away of the spores in the environment because basically there is very, very low or no spores available during the middle winter periods. This information also allows us to understand when we should apply our pruning good protectants, protecting these pruning wounds. So for those growers pruning late, getting into the start of the spore release, there is no doubt that those pruning wounds are going to need to be protected. So not other really comprehensive work on trunk disease has been conducted in other provinces. There are 3 main studies that they were actually including some samples collected from Ontario and Quebec, but these studies were more focused on the status of trunk disease in Northeast United States. And I've been lucky to work with Dr. Wendy Mcfadden, who actually she's actually quite interesting to understand and to know more about the status of this disease in Ontario. So in the last years, she has been able to collect samples that we have been processing in our laboratory. And so far, we can confirm, the presence of all these different grapevine trunk disease fungi in Ontario, which is not surprising and is very common as happening in other grape growing regions. So finally, we are going to move into the management of grapevine trunk diseases, and we'll go through the different steps of chemical, biological, or cultural practices. So first, it's important to say that there are currently no products, chemical or biological, capable to cure infected vines. As I mentioned early in the talk, we have few chemicals like sodium arsenite or the systemic benlate, Venomil, that they were able to fight the infection inside the plant. However, those are either phased out or they are not available anymore here in North America. So basically, the only way we can eliminate the infection in the plant is by what we call remedial surgery. And this is a very clear example provided by one of my colleagues in Australia, Dr. Mark Sosnowski. So basically, we are following the infection and what we call the chainsaw solution, start cutting different parts of the infected trunk, cordon, until we see healthy bud. We usually go about 10 centimeters below the observation of the healthy bud, because we know that the fungus can still be present in this area, even if it's not showing symptoms. And usually use suckers, to retrain those vines and keep production. So this technique it can have different advantages or disadvantages. We can still retrain, the clone if it's an old variety or clone that is not longer available. We already have a very well-established root system, so it's I wouldn't say much easier, but faster returning to full production. And of course, it's much successfully the vines are self-rooted, because once this infection has gone through the graft union, basically we don't have any other option. So disadvantages is very labor intensive, and you need high skilled workers, to conduct this work. There was a study in California done that, a little bit over $2,000 per acre to conduct this type of work. As I show you here, this is a vineyard in California where they did retraining in the entire 150 acres block. And better results if done during the dry period season. As I mentioned before, that's when in California, there were no spores in the environments. You have a much less risk of infection in your new pruning wounds. However, it's very hard to do this work in the middle of the growing season. Usually this work is done in the winter, while pruning and while you have your pruning crews. So that's why control of grapevine trunk disease is primarily based on prophylactic practices, and is right now mostly focused on pruning wound protection. So we have actually different products, chemical or biological, registered against SCA, Utaipa, Botusphera, Dieback. However, very unfortunate for us in Canada, there is still no product registered for grapevine trunk diseases in Canada. We have two types of products. We have paints and pastes, as we can see here in an example. They are very effective, and even more effective if a fungicide is incorporated into the paste, because we not only have the shield protection that the paint will provide, but if we add a fungicide, we will have also this against the spores landing in those pruning wounds. However, they need to be applied by hand, and as you can imagine, they result in very high economic cost. There are still wineries and vineyards that they use paints, but since a while, the industry really is asking and requesting solutions that we can do with a spray application, as shown here in some of these pictures. So we have the possibility to treat large acres in less time, of course. It's much cost effective because we have the possibility to have several sprays, and as I showed you before with our epidemiology and the spores present during the growing season, we may need to go to more than one spray, same as we do with powdery mildew or botrytis of other diseases. But we need information, at least here in Canada, to register or expand the label of some products, and we need also to develop the best application methods. Usually, right now, the recycled sprayers have done very, very well, very good results on applying these products. So this is just an example of the different chemicals that are registered in a place like in California. And so they have many different chemicals or biological products registered against grapevine trunk diseases. One of the things I have to highlight is the difficulty we have here in Canada to bring some products or to get companies interested in registering products is basically an offer and demand situation. If we see California with, over 500,000 hectares versus 15,000 hectares in Canada, that can be an economic impact on registering a product. However, that's what we are actually working in my laboratory to try to, develop these results, get these results, so hopefully we can get some products soon registered in Canada. So in that end, we have been focusing on both, as I mentioned before, trying to develop new biological controls as well as, provide results for chemicals. The main reason for the biological controls here in British Columbia is a very high demand by the industry. About between 15–20% of BC industry right now is farmed organically. There is a general idea, of course, of the sustainable grape production here in British Columbia. So one of our first priorities was to try to develop these biological agents. I have a great master student that has been with me 2 years. The first thing we have done is trying to identify potential biological species. So right now we have identified up to 8 different species in the genus Trichoderma. Trichoderma, we know, has antifungal capacities. So we have identified different species that we have been both putting into in vitro control greenhouse as well as field trials to assess the capacity of these different strains against plant diseases. So what I'm gonna show you here first are the control results for our field trial in 2019-20. So this is our inoculated controls, and basically what we did was pruning different vines in that vineyard, inoculate with the pathogen, artificial inoculation, either 24 hours, 7 days, 21 days, and 60 days. And this, what it's telling us is, even after 60 days of pruning, that bone is still susceptible. Susceptibility significantly decreased over time since we prune, but even after 60 days, this bone is susceptible. So if we have this together with the data on the availability of inoculum, we see that our pruning wounds may be still, susceptible for a couple of months under our conditions here in BC. So these are the results of the products we have used, and here we have 3 main biological control products from British Columbia, and what we did was comparing to 2 commercial products, Gilsil, which is tebuconazole, and T77, which is a biological, Trichoderma atroviridae, and we wanted to see, how our local biological control products stand against them. So here we have the mean percent disease control, so basically the longer the bar, the better control. And again, what we did is after pruning, we were applying the different products and we were challenging those pruning wounds with up to 2,000 spores of Neophysium parvum, which is one of the most virulent. We wanted to see how long, the activity of the product has on the pruning wound. So for example, for Gilsil, we have almost 100% control during 21 days, very good results on protecting these pruning wounds, and decreased, after 60 days to 80%, but still very, very good control. T77, our biological commercial product, really, really good performance. We can see here that over, between 80 and 90%, very consistent through 60 days. And the good news were also that our local biological control agents were able to give us up to, more than 90% control, even after 60 days. So there is very promising results regarding the use of the development, continued development, these biological control agents. So finally, to finish the talk, I would like just to mention how we can manage as well the pruning. So, and this is basically how we can manage the pruning to minimize the infection. I mentioned before, based on this type of data, in California, for example, it's suggested to delay the pruning as much as we can, because when we delay the pruning, we enter in this dry period in California where no spores are present. However, this is not going to be possible in our conditions in British Columbia, because if we are delaying our pruning, which is actually not delaying, is when actually most of our pruning happen, we are actually pruning, when most of the inoculum is in the environment. So our question was, what would benefit pruning earlier in the season in British Columbia? So in order to do that, we have here a Chardonnay block that we prune at different times of the season, in January 15, February 15, March 15, and April 15, again, the following year. And basically, we came on April 16 in both years, so one day after we did our last pruning, and inoculate those pruning wounds with our plant disease pathogen. So we can see here that pruning in the first year, in January, we're able to reduce up to 80% infection. We didn't see such a good results the following year, but still, pruning in January in the second year, we're able to reduce 40%. So compare, we're pruning, done in March, our wounds are still very, very susceptible. So basically 80% of them are infected, or even pruning in April, our pruning wounds are gonna be the most susceptible to infection. So we had some positive results that we could manage, the pruning to reduce the infection. So as conclusions, management of plant diseases, again, needs to occur from nursery to vineyard. It's very important to have these diagnostic tools, try to understand the health status, improve the health status of our planting material, determine the stressors and how these biotic or abiotic factors are going to affect disease development. Try to really understand and avoid any type of stress during the first years of planting. We need to understand epidemiology at the regional level, and I try to highlight how important, is to identify this high or low risk of infection throughout the year. What happened in BC is probably going to be different of what happened in Ontario, and is definitely different of what's happening in California. So it's very important to do that at the regional level. Need of long-time protection of pruning gums. As I mentioned before, there is promising chemistry in the market. We should not underestimate biological products. I think we have shown very, very good results, giving us up to 60 days protection. And by managing the pruning, we can reduce, with early pruning, in some cases, up to 80% of infection. But very important, vineyard sanitation and control applies since year one. And this is going to be critical if we want to have success in managing these diseases. As I mentioned before, we need to manage trans diseases as we manage powdery mildew or as we manage botrytis. Since year first, we have to be on top of it. This is just a review paper we published in 2018, where way more of what I presented today is available. It's free for download in plant disease. So it's basically a review of most of the, or all of the current control strategies available against trans diseases. So finally, I would like to acknowledge, of course, our funding sources, the BC Wine Grape Council, Agri-Food Canada, and CGCN. Funding from some of our industry. None of this work would have been possible without my lab and the people working in my lab, as well as all the collaborators that I have, supervisors and committee members of the different students. So I thank you, all of them, for being part of this project. And that's all I have for you. Thank you very much.
Okay, well, thank you, Jose. That was some really fascinating work that you've been doing out in Ag Canada at the Summerland Research and Development Center out there in British Columbia. We're now going to turn to the Q&A portion of the lecture. We got quite a few questions, which is fantastic. So everyone just settle in for a great discussion here to come. Our first question, directed to you there, Jose, is from Debbie, and she asks, are any of these trunk diseases more prevalent in spur pruned or cane pruned vines?
That's a great question, and it's something that we are still trying to, I would say, scientifically prove if there is any difference. What we can see based on the research we have is we see trunk disease symptoms earlier in a spur prune, than cane prune. If we have those vines with the cordons and the spur positions, because of the way trunk disease work, we are going to see those infections. They are going to be detected earlier in that portion of the cordon, in that spur, that is in a spur prune. In cane prune, because most of the work is done at the head of the vine, and most of those spur positions or cuts are going to be at the head of the vine, we have infections, but those infections are really entering directly into the trunk, which means that we are going to have way, longer time to detect the infection, because, of course, the trunk portion is a much bigger portion, much wood for the fungus to colonize, and we have seen those experiences. We have seen regions where usually cane prune has been done, and I've been told, no, we don't have trunk diseases. My, cane prune vines are perfect. Those vines start getting into an age of 20, 25 years old, and they start suddenly collapsing. What is happening? This fungus on the trunk has basically, it takes that long to colonize the entire trunk, has colonized the entire trunk, so those vines literally on cane prune collapse. While if you have this spur, cordon prune, you are identifying the infections earlier before they actually get into the trunk. So both get infected, because in both type of pruning we are doing pruning wounds. But I think that's the main difference we can say now, is we are able to detect the infection much earlier in, spur prune cordon vines than cane prune vines.
Okay, thanks. Our next slew of questions, we have a couple here from Wendy Mcfadden Smith, who's actually our speaker next week. So her first question, so Wendy's first question is, when presenting vine age and trunk disease development, is it the whole vine age or trunk age?
Is the whole vine age. So we are basically calculating with the year the vineyard was planted. is the total vine age.
Okay, and her next question, can cold injury, as in trunk splits, increase trunk susceptibility to trunk disease?
Yeah, this is a great question, and this is something that we should definitely address, here in Canada as part of our, climatic conditions. So one of the things I observed in British Columbia while doing our surveys is that younger vines, you can see much bigger cankers develop in younger vines, primarily because what we think is happening after these cold winter events, you get those splits. So you have a much larger surface area that can get infected by the pathogen, or the spores can get into that. There is no doubt there is a higher susceptibility to the plant to not only trunk disease, but other type of diseases, when these splits happening. Mm-hmm. One of the things we have with these splits of this spray is we usually see earlier crown gall symptoms than trunk disease symptoms. So a lot of these plants that split and they have these cracks on the trunk, they are usually going to be going down earlier because of crown gall and trunk diseases. But so to answer Wendy's question, I would say yes, when the trunk is split, we have a higher surface area that is exposed to the environment. So there is a higher possibility that spores are getting into that large area and getting infected. And I think it can have a abiotic factor, a stress factor for the vine susceptibility. However, my experience tells me that most of these plants are dying before or many of them for crown gall than for trunk diseases.
Okay. What would you say is more optimal for infection? Would it be a few large cuts or many small cuts?
So that's a great question. And there have been some studies done. So usually what we recommend is to avoid any unnecessary cut in the plant, of course. Any cuts that you're going to have a much larger cut, if you can avoid it, sometimes you cannot avoid it. It's just a chance, it's a chance that the spore lands on your pruning gun. If your pruning gun is larger, your cut, you're going to have a much larger chance that the pruning gun lands in that area. That if the pruning gun is smaller. We also know that large cuts, large pruning guns, takes much longer to heal than small cuts. Small pruning guns, like a thumb or a dime, they will heal much faster than a very large pruning gun. Mm-hmm. So they both can get infected. Large cuts are going to be susceptible much longer time than small cuts. So some of the operations we do in the vineyard, we cannot avoid to do these big cuts. Mostly, sometimes you have to reduce these big spurs position. You have to reduce them, cut them, develop new spur positions. So some of them, they cannot be avoided. But in those cases, for those big cuts, I would really recommend at least, to have a paint or some kind of paste that can protect this big, large cut, that it will take much longer to heal and is still susceptible for a long time.
Okay, great, thank you. So we're getting through them, quite a few left to go. So the next question is, would Senator work the same way that Benomyl used to? The same fungicide family, and it's gone through PMRA review.
Senator?
Senator, yes.
I believe Senator is tebuconazole. I don't think it's the same component that Benlate or Benomyl. It's a different active ingredient, I believe, if I have correct the. So the main difference between Benomyl, Benlate, and the fungicides we have right now, Benlate was a systemic fungicide. So Benlate or Benomyl will, systemically interact with the infection inside the plant. Most of the fungicides we have right now, like Senator, which I believe, if I'm right, Wendy's going to help me there, is a tebuconazole. They are all protective. They are all they will not go systemically. So that's why we are using in pruning gun protection. They will eradicate the spores landing on the plant. That's why we currently don't have any systemic fungicide that I'm aware of, and to the best of my knowledge, we don't have any systemic fungicide that had the same capacity that benlate to fight against Trund diseases.
Okay. Thanks for that. The next question comes from Alex, and Alex wants to know, what are the specific use recommendations for pruning wound compounds, Time window after making a cut, is it okay to apply if the wood is damp? Is effectiveness temperature dependent? Any better for some varieties over others, et cetera, et cetera? A lot to kind of unpack there, but I think he's just looking for overall specific use recommendations.
Yeah. So there is a lot of things and a lot of factors that can go into pruning wound protection. As I mentioned before, if you are in a region that you have a good idea of your presence of inoculum in the environment, like I saw here in British Columbia, for example, here in British Columbia, growers pruning maybe December, January. If they don't apply any pruning wound protectant to that wounds, they are going to be very safe because we know that there are no spores in the environment. It's too cold. And also, the other difficulty we have in many of these regions is some of these paint pastes are frozen. I've seen that they cannot apply because they get frozen. So it's very hard to apply in those situations. For example, if growers are here, and I'm talking for the experience I have in BC, and because we know when the inoculum happen in BC, if growers are going and pruning closer to where most of our inoculum is happening, which I would say March, April, a lot of pruning is still going here in March and April. There is no doubt that my suggestion, my advice would be to treat, as soon as possible after you prune. We cannot control the weather. We don't know when we are going to go above freezing temperatures. We don't know when, we have certain knowledge of when a rain is going to come within the 7-10 days. So we have this room, to play with, but overall, as soon as you prune, if you are in this window and in your region you are under higher risk of infection, you should treat your wounds. What is the main problem we have in Canada? We still don't have a product registered against Trund diseases, and this has been kind of my battle, to try to get industries interested in registering products, in bringing products to Canada, and even conducting, some of the results generation, for these companies to be able to register. So hopefully in a short future, I mean, we all know how this goes in terms of registration, but hopefully in the next years we can see products coming into Canada for pruning wound protection.
Okay, so we'll be expecting an update from you on that when the time comes. The next question here is whether it's safe to replant into a vineyard where trunk disease was a problem. So can the fungi survive in the soil or in root fragments? Can pathogens be transmitted between adjacent vines via root grafting?
So that's a really good question again. So we have two different situations here. As I mentioned in our source of inoculum, we can have one of the pathogens we have on Trund diseases is the blackfoot pathogens. These are soil-borne pathogens. So these cylindrocarpon, Ilionectria, they are going to be in the soil. So if you have a vineyard block that has been, heavily infected with cylindrocarpon, if you pull out that vineyard and you plant new vines, the pathogen is still going to be there. And we have some data that up to 3 or 4 years, this type of fungi can survive in the soil. So it can be a risk to that, definitely. In terms of the risk of the pathogens, one of the main complexities we have with Trund diseases is not only grapes. We can have spores coming from many different hosts surrounding. Here, for example, in BC, we have identified from cherries, from pears, from plums, native plants. We have a very large range of native plants surrounding vineyards, cottonwood, ash trees, willows, that they are also hosting those diseases. So it's very, very hard to completely avoid, the presence of infection because of this situation. However, the more sanitation and the cleaner we are, the best we are going to be trying to fight these diseases. I mean, there are things we cannot control. I mean, we cannot go now here in BC and decide to all the black shades be removed because of Trund diseases. It's not going to be feasible to do that. So we have to do the best we can in our vineyard or within our vineyard to that. But yeah, there is a possibility that that this particular fungi, blackfoot, can be in the soil. Fumigation would be a possibility, but we know currently the situation, with fumigation, the cost, the environmental concerns is very complicated. Yeah, unfortunately, I don't have a really straight answer. Root grafting, we don't have any evidence that these pathogens are transmitted by root grafting, as for example, viruses would do. Basically, by the time this pathogen will get to the roots, any of these cankers, the plant basically would be dead. The trunk would be completely colonized. So we don't have any evidence that root grafting would be the main spread of these pathogens.
Okay, thanks for that. It's a lot to work with anyway, even though you might not have had a straight answer, as you mentioned. There's a lot to unpack there anyway, and a good answer. So we're working our way through, still a few to go. Our next question comes from Amy, and Amy asks, does hot water treatment of dormant bench grafts reduce the number of viable fungal spores?
That's a fantastic question, and this is something that European groups started to look into that a few years ago, and we, luckily we have some data on that. So unfortunately, the results from that data says that hot water treatments, at the way we do hot water treatments, 53, 55 degrees for 30 minutes, that's kind of the standard to eliminate other type of organisms that we target with hot water treatments, will not totally eliminate Trunk diseases. Some of the problems we have is if we see where the infections happen in the wood. There is, if we have a cutting, go through hot water treatment, it may be, 1.5 centimeters diameter. Some of these fungi will be in the middle, in the pith even of that vine. So getting those temperatures all the way, even we think is a small diameter, is very hard. It's very hard to get those temperatures for the time, that they will need to eliminate. So they saw that some of the pathogens on the surface, or very, very close to the edge of the surface, they can be affected by hot water treatment. But I would say I will not rely on hot water treatment to fully eliminate Trunk diseases from the plants. Hot water treatment has been designed, or it was designed to eliminate viruses that they are flow limited, so they are actually very, very close to the edge where we can reach those temperatures, or insects that are actually on the surface, outside on the surface. So those are what hot water treatments. It was not designed originally for Trunk diseases, so that's one of the issues we have. Okay, however, said that, I think it's very important to highlight that our studies and our research indicates that we believe that some of these fungi may be even part of the microbiota of the organisms in the vine, and that's what I've been showing. They may be latent. They may not cause any problem to the vine until the vine gets in stress. So that's why we think that some of these fungi actually they may be part of the general organisms of the plant. So this is very important to take into account, because when we go to clean plants, eliminated plants, well, what really is a clean plant? How much we want to clean a plant? It's going to be completely sterile plant with no microorganisms inside. It's another subject, but just wanted to bring this up.
Yeah, sounds great. Sorry, I started talking over you a little bit there because I didn't realize that you weren't finished. Apologies. So our next comment is actually kind of interesting. So it comes from a gentleman named Bill Eggert. You actually did some work for him. He says you came and took trunk sample in a vineyard he managed that showed symptoms. He wanted to provide you with an update. He found a marked reduction in dieback by reducing irrigation. The reason he brings this up, he says, is because you were talking today in your lecture about how water stress can enhance this issue. So he just wanted to ask if you could comment on the difference between the work you did there and maybe what you're talking about today.
Yeah, no, this is great. Yeah, I remember perfectly, exactly. Mm-hmm. So, I mentioned water stress as one of the main targets we have as an abiotic factor that may cause stress in the plant. This doesn't mean that water stress is the only one or is the correct one, that we still don't know. It has been some research actually in Australia where they have shown that they see higher symptoms on Eutypa dieback when vines are overwatered than when they are with water stress. So this is a very, very interesting result, which actually kind of correlates with the observations that we have here. So it's very new, this area of research, but when I mention water stress, it's basically, you know, we are start targeting some abiotic factors that we believe it may cause some stress, but we should probably also look to overwatering, you know, as another factor that that they may cause stress. So definitely, we don't have the answer yet, but yeah, I had to say that the observations correlate with some of the current research we have coming from Australia.
Okay, great. Hope that answers the question for you there, Bill. The next question comes from Wendy again. She asks whether pre-pruning makes vines more susceptible.
Pre-pruning.
Or could it?
I don't know, honestly, Wendy. I don't know if they could be vines more susceptible. I mean, I don't know if she mentioned pre-pruning as having a first pass, you know, if we have a BSP trellis, so many people do a pre-pruning to try to eliminate a lot of the pruning debris, and they go after with a final pruning. I don't know. I mean, we still have to focus in the final pruning we do. The final pruning we do, those wounds are going to still be susceptible at the time we prune. So I may have to go back and forth with Wendy in some emails, so she can clarify me what she means with this type of pruning.
Wendy might very well have commented in the chat as we were talking about it just now. So as I get through the questions, I'll take a look back, Wendy, and see if you've clarified for Jose there. And if not, you guys can follow up afterwards on that. So Diana has a couple questions for you. We've got, looks like about 10 left. So we're getting through there, but people are asking a lot of questions here, which is great. So Diana asks what pruning wound protectants are available and work on trunk diseases. I think you talked about that a little bit, but I'll ask even still just so you can comment.
Yeah, so there are many of them available. Unfortunately, we have none registered in Canada, and that's the main problem we have here in Canada. And that's where most of my research is going towards, trying to bring some pruning wound protectants, you know, registered in Canada. If you see some of my slides, I put, for example, just example in California, there are many products they have registered, you know, they have myclobutanil, they have thiophanate-methyl, they have biologicals with Trichoderma, they have BTC, which is a polymer, actually, is produced in organic vineyards. So the tebuconazole has done really good results, you know, and it's one of the main products used in Australia and New Zealand for pruning wound protection. There are a lot of products there. Unfortunately, we don't have any registered in Canada. That's our main, I guess those are the homeworks we really need to work on and bring growers, you know, product registration. So yeah, I'm sorry that we are still few steps behind other places, but we are getting there, hopefully.
Okay, so the next question comes from someone who's out in BC with you. Her name is Lisa, and she asks where growers can best submit samples for GTD analysis out in BC. She mentions the Ministry of Agriculture's Plant Health Lab in Abbotsford, perhaps, and she also says she assumes Sur DC has limited capacity for this outside of your research.
Yeah, so exactly here in British Columbia, the provincial lab, the Abbotsford provincial lab, they have the mandate of extension, and they have actually the laboratory for service. So samples can be sent there. I work very close with the pathologists there. They are great pathologists, Siva, Vipen. So sometimes when they receive some of these samples, because they are used to receive maybe different type of samples, you know, they are in the lower mainland, so they work a lot with blueberries or other type of. I work very close with them, and sometimes, you know, even we commented on the samples and everything. But that's the best place in BC to send the samples. Within my research, because it's part of my research, I've been actually contacted by growers, and growers have called me to take some samples, and because I can use them as part of my research, I've been assisting growers with that. However, unfortunately for us in Canada, we don't have a service lab. My lab is not for service; it's for research. So I'm very limited on the work I can, you know, the service I can provide. But within my research, it's hard. But I mean, I never try to say no to a grower, and they understand how busy we are. But yeah, between the provincial lab and I, we can handle samples. And Wendy has been sending me samples from Ontario as well to take a look to some of the blocks there.
Okay, thanks for that. I also believe you might have touched upon this in one of your past answers as well, but I'll ask it and you can just kind of glaze over it if that is the case. So Amy asks whether any of the chemicals identified by Dr. Sosnowski as effective pruning wound protectants are registered for post-pruning application in Canada.
No, unfortunately not. As I said before, we have not a single active ingredient, whether chemical or organic, biological in Canada, to the best of my knowledge right now. I mean, I have to be careful with that because of course I don't know everything, but I don't think there exists any. But for example, some of the products he is been working and showing really, really good. Tebuconazole is one of them. So that's one of the products I've been generating data here in Canada. So hopefully the company is interested and going through a registration. Some of the other products they have been working there, we have been testing them here. I've been working a lot with products that we have currently registered for grapes. So for other diseases, like for powdery mildew or botrytis. So the idea is if any of these products also work against strand diseases, it's much easier to have a label extension than a completely, registration of a product from scratch. So we have some of those in the pipeline that we have been testing, and we have some results. Unfortunately, like these things, the best products that they work against strand diseases, we don't have them registered. So the company will have to go for registration, full registration.
Okay, and just to add to that, I see that Wendy commented, and you kind of touched upon this a little bit too. Several of the fungicides are indeed registered on Grapevine, just not for trunk diseases.
Exactly, yeah, exactly. I should have clarified that. So we don't have anything registered for trunk diseases. Some of the active ingredients and for example, myclobutanil, which I believe here in Canada is called Nova, is registered in California for trunk diseases. We have it registered here for powdery mildew, but not for trunk diseases. So okay, these are some of the products I've been doing some work here in Canada to try to see if they can be some label extension as well.
Okay. Another question we have here is, what effect would fungicide sprays for other diseases, so downy, powdery, bunch rot, have on the biocontrols? Wendy has asked.
That's the next step of hopefully my next master student, or that we can have. We still don't have data produced here, but there is definitely, if we are using biological organisms like Trichoderma or some of the bacillus or some of other fungi, we know that some of these chemicals can affect them, and they can reduce the control of these biological control agents. So we need to get more research on that. There's some research from Europe mostly where they have been really working on biological control. But yeah, there is no doubt that even sulfur, we are even trying to see the application of sulfur has any impact on Trichoderma, for example. Because if you are organic grower, powdery mildew, most of your control or one of the only options we have is sulfur. So we have a lot of growers just using sulfur. So just because it's organic doesn't mean that it's not going to have an impact on another fungus. So we need to look into that more in detail.
Okay, so working away here, I still see a few more. So hopefully everyone is still with us. The next question here is from Bill again. So he asks if the temperature differences in the 2 years of pruning time study were noted. So was it a warmer year, earlier bud break in 2018? He asks.
That's a great question, and I'm hoping to answer that question in the next few months. We are finalizing, analyzing all the data. We still don't have that correlations on that data, but it could be easily that, or we could hypothesize that different climatic conditions, they took longer, these pruning wounds to heal. That's why they were more susceptible. But that can be one of the main reasons. We still need to analyze the data. We are hoping to get that work published this year, actually. So hopefully it's going to be available for everybody. I just gave a snapshot of the work.
Right. Okay. Greg would like to know how he obtains the local park biological products. Does he have to apply them with pruning paste?
So right now, what we are doing, because we are, of course, in the development of these biological agents that we have here locally, we are applying them with a spray bottle. I mean, we are conducting experimental trials. We are not having like a commercial trial going. So we have concentrations that we usually put 50,000 spores of the Trichoderma species on the pruning gun, and we use basically a spray bottle. There are actually biological control products, like for example, the one we are comparing, the commercial, the T77, that they actually come, they come in a dry powder that you have to, dissolve in water to apply, and those products can be, apply with a sprayer or, so the idea is to have all these products to be able to apply with a sprayer to basically minimize the cost. I mean, it's very costly to apply with a brush or every single pruning gun. It can be done, but it's very, very costly. So all these products are meant to be developed into sprayers.
Okay. And I look like I see about 5 more, and that's probably all the questions we'll take for today. But we'll see how we do after these ones. But the next question is, how do you balance pruning early for trunk disease with pruning later after winter damage? So Tom wants to know how many buds to leave.
Yeah, exactly. And this is, with my background coming from California, sometimes I also have to learn, that growing grapes in a cold region is quite different. So of course, pruning earlier may have other disadvantages, that the vines maybe is my understanding that if you prune earlier, they may be more susceptible to a spring, frost or winter injury. We haven't done these results. Based on observation, pruning earlier in our research blocks didn't impact at all during these 2 years, the pruning weights or the crop that we obtain. But of course, every year is different, and we will have to go and look into the temperatures. Maybe leaving an extra bud in that if we are pruning earlier, and instead going to 2 bud spurs, leaving 3 bud spurs could help that. But I'm not a physiologist, I'm not a viticulturist, so they may have another implications, so I understand it's complex. I'm approaching the pathogen disease aspect, but that's why, I work with great people in the center, like Carl Bogdanov or Pat Bowen, who are actually plant physiologists and viticulturists that sometimes they bring me down to reality. Yeah, you can do that, but if you do this, there may be other issues so we are trying to put all this together and to work on this together. We don't have a final answer yet what we really mean to prune earlier, but that's some of the disadvantages you may have. Yeah.
Okay. And when you're removing a trunk, is it best to cut 3 centimeters from the graft or 30 centimeters from the graft? Phil has asked.
When you remove a trunk, meaning, so if the trunk is infected, you want to retrain the vine, I assume. So so I guess the question is how far away from the graft you cut the trunk. Well, if the trunk is clean, there is no infection, close to the graft union area. Usually we usually advise to cut 10 centimeters farther than the last stain we see, the last infection we see in the trunk. So if we start cutting the trunk and we are seeing canker going, once we don't see any canker or any type of symptom in the bud, we still advise to cut another 10, 15 centimeters extra from that last symptom, last stain, because the fungus can still be in that part of the bud ahead of the symptom, but it takes time to produce this canker. So I think it's maybe not so much how far you can cut from the graft union, but how much room you have from the end of the symptom on the stain in the trunk. I don't know if I made myself clear.
Okay, well, Phil chimed in and said that was indeed what he was asking, so you did address the question that he wanted to hear. Yeah, thank you. Okay, we have a question here. So Jadran wants to know if we were able to find a correlation with nursery material thresholds of infection and disease development in the field. Is there any possibility to have nursery material certified as free of grapevine trunk pathogens?
Okay, so we are actually conducting experiments to answer this first question. So all we have done right now is with the molecular tools we have developed, we have been able to establish that we know that there are different pathogens at different concentrations happening in the plant material, in different parts of the plant material. We haven't been able yet to assess what that really means when you plant those vines. So right now, my PhD student is conducted in both greenhouse and field experiments where we artificially inoculate cuttings before we root them. So we basically get canes. We artificially inoculate it inside with a vacuum system we have. So we are inoculating with different concentrations of the pathogen, from 25,000 spores to 5,000 spores to 1,000 spores. So then we have a threshold there that we know exactly how much pathogen is in the cane. Those canes, what we do is we root them, and then we plant them in the greenhouse or in the field. And those plants are right now going under different type of stress conditions. So we have a water stress experiment, we have a nematode experiment, we have a gel rooting experiment. So I will come back hopefully in 2 years and answer that question, depending on the threshold we have, and depending the type of stress we have on those plants. So the hypothesis is that plants with less amount of spores will be able to, fight better this infection, and plants that they have, high number of spores and are under stress conditions, they may develop these symptoms much earlier.
Okay, great. And I just want to check in with you, Jose. How are you doing for time on your end? We actually have a few left, and if you're good to stay, we'll keep going through them because we've got a lot of questions here. People are still sticking with us. If you're good to continue,
so the sun is shining, so I'm eager to go out to take some sun. But no, I'm totally fine, totally good. Yeah.
Okay. Yeah, I can see the color change in your room from the light blaring in there. So. Okay, just a few more. Thank you so much for your time. We really appreciate it. I know everybody in the chat here today appreciates it too. So would training a new trunk be a good way to improve the vine if the shoots are showing symptoms of trunk disease?
Can you repeat, please? I don't know if I understand the question. When retraining
Yeah, so would training a new trunk be a good way to improve the vine if the shoots are showing symptoms of trunk disease?
Okay, so basically I come to what I answered before. So renewing vines, so having a new trunk, retrain, if this is what I understand from the question, is sometimes the only way we have to keep the vine, to remove the infected part and keep the vine. If we want to keep those vines of that vineyard, that's the only way we have to do it. Some people, what they do is they start with 2 trunks, in some areas, not specifically for trunk diseases, mostly for other issues like winter damage or crown gall. They have 2 trunks going all the time, so they are able to cut one. They still have one producing. If the other one gets infected, they have time to retrain the other one. I mean, growers know what is that. I don't know if I understand correctly the question, but if the shoots show symptoms of trunk diseases, again, it's the same thing. We have to continue looking into where this canker goes through the vine, through the cordon, and through the trunk. And if we want to retrain a new trunk, we will need to eliminate entirely that infected part to have a new trunk. So the new trunk coming from a water shoot from below the infection, it should be okay. It should be okay to have free of trunk diseases. If I understand correctly the question.
Okay, thank you. And so we're going to call it at 3 more questions here, and I haven't seen any more come in, and then we're going to be all done for today. So Steve has asked, New Zealand has some success with, and apologies if I butcher this term here, Steve, Trichoderma treated dowels being inserted into Eutychia infected trunks, EUTYPA, infected
Yep.
trunks. Your thoughts on this, Jose?
Yeah, I know of the work, and it has been a work that they have been conducting in New Zealand to try to get the trichoderma going and expanding inside the trunks. It has some very good results. They were able to get the plant colonized with trichoderma. It's internal, so the trichoderma is able to fight the internal infection against eutypa. So there have been some successes. It's one of the techniques that they have been trying to develop to get a longer term. Instead having to apply the trichoderma every single year in the pruning wounds, trying to have the trichoderma colonizing the trunk or the plant. So it has tons of success. It's costly, I mean, you have to go for every single plant doing this type of work. If you think about, drilling every trunk, it's costly, and we also have to think that we shouldn't forget that trichoderma, I should have mentioned that, is a soil inhabitant organism. So trichoderma, the natural habitat of trichoderma is in the soil and in the rhizosphere. That's where we find most of the trichoderma. So we are using, because these fungi have a very high antifungal capacity, but we are using a soil inhabitant to put it on an aerial part of the vine. We are getting exposed these fungi to light, to relative humidity, to different temperatures. So there are many other different variants that that's why trichoderma may not, may work differently. So, but I'm aware of the work, and if the grower can absorb the cost, we have a project in California where we are actually drilling, and injecting a specific systemic compounds. They were showing very good effect, but as soon as growers were making the numbers, how much will cost to apply that into a full vineyard, that's where, the reality comes. So unfortunately, that's another thing that growers have to balance.
Okay, and I believe, Deborah, that answered your question a little bit in there. If that's not the case, just go ahead and ask in the chat. You were the last question that I was going to ask today. She also mentions trichoderma is also plant endophyte, if you wanted to comment on that to finish it off.
Yeah, so exactly. So we can find trichoderma in, as an endophyte, meaning they are natural part of the organisms in plants, including grapes. Actually, all the work I shown with trichoderma, those trichoderma were isolated from grapes in British Columbia. So we were getting them from the roots, from the basal end of the trunk. Trichoderma happens naturally in many plants endophytically. So one of the ideas we have is trying to see which type of a species we can adapt better to this pruning wound, so they are able, hopefully, to endophytically colonize and, be present in this aerial part of the vine. So hopefully, and this is some of the work we are doing right now, applying trichoderma this year in the pruning wounds, coming next year, and when we prune again, if the same trichoderma is present in these pruning wounds. So this is some of the work we are doing now. It's endophytic. We know that they will colonize without causing any harm to the vine. But again, we are getting a soil inhabitant to put in an aerial part of the vine, which is a completely different environmental conditions. And I think that's one of the things that we see a significant reduction when we inoculate pruning wounds. We come back the year after, and we see a significant reduction of finding again the same trichoderma on the wood. But this work is ongoing.
Okay, and would a hand application be cost effective if it's not an annual cost? So Deborah just wanted to follow up about the trichoderma and control products, and whether you anticipate having to apply it annually, and whether that annual application could be cost effective.
Yeah, so for example, one of the things we have seen with trichoderma is giving us a much longer time control in this pruning wound. So we have done experiments up to 60 days. And even after 60 days of applying the product, the trichoderma, we still have very, very high, because the trichoderma is still, on the pruning wound, it's still viable, it's still growing, versus some of the active ingredients, chemicals, they will degrade on time, So with time, that's why we may need to reapply some of these chemicals if we choose for a chemical pruning wound protection, because it will have maybe these 7, 14, or 21 days efficacy. With trichoderma, maybe one application of trichoderma would be enough. because we have a much longer term. So if you apply by hand, depend on the size of your vineyard, and depend. I mean, this is everybody's, a different situation. Your numbers, it may be feasible. I know many people are still, applying by hand some of these products, and if we only have to do one application, but the thing is, if we still can apply these products on a sprayer, having the same result, whether it's one application or two, having a sprayer will also minimize the cost. So the beauty of these products is we can easily apply with a sprayer. So we don't really need to do the hand application as we used to do when we have more on the paints and the paste, that those ones really we had to apply by hand.
Okay, fantastic. Well, that's all of our questions for today. Thank you so much to everyone for contributing to such a lively and informative discussion. And thank you so much, Jose, for sharing so much of your time and your expertise with us today. I know I learned a lot, and everyone here that participated and listened in has as well. Just to finish off, if anyone has any further questions or wants to contact you, Jose, what's the best way for them to do that?
Through my email, through my Canada.ca email. I mean, if you just Google my name, it's it's an easy name to find on the internet. It's a long one. So José Ramòn Urbez Torres at Canada.ca. They can go into the Summerland research station. All my information is there. So yeah, they can send me an email, and I will be happy to answer as soon as I have time for that. So that will be a problem. Yeah.
Okay, great. And just so everyone knows, I think I mentioned this already, but just a reminder that this presentation will be posted on our COVI website, which is brocu.ca/COVI. We'll be able to rewatch this there and learn more about the work that Jose is doing. Thanks again for everyone for being here today, and I hope to see you all next week. Our next speaker will be Wendy Mcfadden-Smith, who, if you were with us today, obviously you heard her chiming in with some discussion questions throughout the lecture. She's an adjunct professor in biological sciences here at Brock University, as well as a tender fruit and grape IPM specialist at the Ontario Ministry of Agriculture, Food and Rural Affairs. She'll give us an update on her ongoing grapevine virus research on Wednesday, not January 27th, next Wednesday in February at 3 p.m. here in the same spot. And you can find more info on our website and tune in and join us then. I hope you can see us then. Thanks very much, guys. Have a great afternoon.
Thank you very much for the invitation. I appreciate it. Thanks.
Cheers. Thank you.